Ideally, these drugs should be easy to take, a short course of oral therapy or a short course of intramuscular injections, and really not to have significant adverse effects that require monitoring or require hospitalization of patients after the therapy. Current therapies where there's repeat injections and adverse effects require sometimes long hospitalization, which is very difficult in regions where there's a poor healthcare system and patients may have to travel a long way to find a hospital. Again, we're looking for medicines that will be stable in difficult climatic regions, so they should have good chemical stability and formulation stability characteristics. And at the end of this, we're looking for treatments that can be made available to all the patients that need them. So we can't afford to come up with extremely complex and very expensive new molecules. And so we're looking for a low-cost treatment course to really make them available to all the patients that need them. What we think we need to improve on the existing standard of therapy. But let's just have a little think about Leishmaniasis itself and the Leishmania parasite, where it resides in patients, and that the issues that we need to address as medicinal chemists to design molecules that can reach the site of action, kill the parasites, and leave the host healthy. So the Leishmania family of parasites are single-celled trypanosome parasites. They have multiple life cycle stages. After being bitten by an infected sandfly, the parasites will migrate through the body and infect a variety of cells, particularly white blood cells, macrophages, and the target organs include significantly the liver, the spleen, the bone marrow, and some other tissues. But once they're inside a cell, that's not the end of the story, because the parasites migrate inside acidic phagolysosomes. So there's additional barriers for a drug to cross before it can find a parasite. It would have to get into the cell, it would have to go into the phagolysosome where the pH is significantly lower, typically around 5, and then get from the inside the phagolysosome into the parasite to kill the parasite. So there's a number of membranes and a number of pH gradients that a compound would have to traverse to reach the parasite, and that's really summarized in this very simple phenotypic screening slide. So macrophage, inside that a parasitophorus vacuole, inside that an amastigote, so that's the intracellular form of the Leishmania parasite, hiding out, trying to evade our drugs. Happily, the cornerstone of drug discovery for visceral leishmaniasis at the moment is phenotypic screening, and our gold standard in vitro assay is actually an intramacrophage assay, where we measure the ability of a compound to enter a macrophage, kill the parasite, and at the same time we can assess the selectivity for the host cell. So kill the parasite and leave the host cell healthy would be ideal, and we can measure IC50 and CC50 for those two endpoints and to work out whether we've got a good in vitro drug candidate. So by using these phenotypic assays for screening to identify new starting points, but also for supporting our optimization programs, we can perhaps capture or at least capture many of the events that we need to deal with in a single assay. So when we're optimizing for compounds with improved activity, it may not just be improved binding or interaction with a molecular target, but it may also be improved properties for reaching the parasite and reaching that target. It's a bit frustrating that sometimes we tease those things apart, but at least we're able to, if we find a compound that has improved activity in these assays, we know we're going in the right direction. So if we were to sum that up, the translational drug discovery challenges which all drug discoverers face are somewhat different for infectious diseases and for neglected diseases such as this. So if we can find a helpful molecule such as I've depicted here, we're really trying to deliver that to a patient to treat the infection and to help them recover quickly. There are a number of questions for us. Which Leishmania species are we tackling? What life cycle stage is it at? Is it just after infection? Is it an intracellular form? Is it replicating or not? So these are some, if we were to set up different in vitro assays, there are a whole load of different assays we could set up, but at least by using a phenotypic assay with the most difficult parasitic stage to target, that intracellular stage, we can perhaps address a number of these issues in a single assay. That's just an in vitro assay, so really to have an idea of whether a compound can be a potential anti-leishmanial drug in a more dynamic system, we need to use some animal models. So a mouse or a hamster model is not the same as the human disease. It really is just an ability for us to test whether a compound has the right pharmacokinetics to reach and kill parasites in an animal. But because the progression of the disease in those rodent models isn't really the same as it is in human, and the immune response is different, we can't really call them disease models, but they're useful in vivo assays, and they give us an indication of whether we're getting the pharmacokinetics right and the penetration of the compounds right, such that we should then go on and try a human experiment to see whether we have a potential drug. So probably still a lot of work to do for DNDi and our partners to better understand this and to characterize the translation between the different models. And that's a story for another day, but I think we're making some progress there. So really to come to the meat of the talk today, it was to present our target candidate profile and our screen sequence. So having heard some of the issues for the disease and the models and what we're trying to achieve, we've tried to reduce into a single slide here the key criteria that we would want to see in the molecule at the end of an optimization program to make us think that it's worth running the regulatory toxicology studies and considering taking a compound forward into human clinical trials, first in volunteers and then in visceral leishmaniasis patients. So in vivo, we're looking for compounds that will deliver if not a sterile cure, then a very high level of reduction in parasitemia in the tissues that we know to be important in the human disease, so particularly the liver and spleen. If we could remove all the parasites below the limit of detection, that would be fantastic, but even to reduce the level very significantly, a 2-log reduction, greater than 95% reduction in the parasite load, is likely to be useful in humans, is likely to allow the immune system to take over, and indeed many patients that show a good response to therapy go on to have a low level of infection, but they're no longer symptomatic. So you could always argue it's better to have a complete 100% reduction of parasite load, but we're looking to, as quickly as possible, bring forward useful new medicines that will help patients. So we'd be prepared to explore the clinical profile of compounds that hit this 95% barrier. To underwrite that, we'd be looking for activity against not just Leishmania donovani, a lab strain, but a variety of strains and isolates for Leishmania donovani and infantum, but also against drug-sensitive and drug-resistant strains and isolates from different geographical regions. And we'd be looking for compounds that can give a 99% knockdown in an in vitro assay, so really complete inhibition of the parasite, no residual parasites hanging around for any reason at high concentrations. Ideally a sidal mechanism of action, again for those immunocompromised patients, and for a faster response in all patients to directly kill the parasites would be desired. If we can prevent their replication and allow the immune system to tackle the parasites, that may be okay, but clearly to quickly kill them would be more attractive. Whilst doing all of that, we want to leave our human patients, our human hosts, healthy, so we don't want to identify any major in vitro signals that would cause problems with subsequent development. So the routine cytotoxicity assays, HERG assay, gene tox assays, so the AIMS assay, the micronucleus assay, and broad ligand profiling against receptors, enzymes, kinases, transporters, etc. So we'd really seek to weed out and design out any off-target signals as much as possible. And then in vivo, we'd be looking for a compound which has a useful therapeutic index. That's to say, in toxicology studies in 2 species, so well, initially at this stage, it would be a rodent species such as rat. We'd expect to see no significant effects at the concentrations that we believe to be efficacious in an animal model of disease, and ideally this should be at least a threefold split between the no adverse effect level and the effective level, giving us 95 or 100% reduction in parasitemia. That might seem quite a small therapeutic margin, I think for anti-infective drugs, very often the critical piece is all about therapeutic margin. We're looking to selectively kill a parasite without harming the host. Many mechanisms will start to be toxic at higher concentrations, and I think our experience tells us that there are a number of successful anti-infective therapies with quite low therapeutic indexes, and as long as the pharmacokinetics are well understood and the dosing is well controlled, they can still be safe and effective therapies. So this is a starting point for us. Obviously, a bigger therapeutic index would be better, easier to develop, and give more room for increasing dose in the clinic if needed. But this would be a base case for us, and we have done and would take additional candidates forward with a therapeutic index of this size. That's all. To have an effective and safe medicine is all very good, but we need to be able to synthesize it, formulate it, and dose it by the intended route of administration, so orally or intramuscularly, and that should be a short course, so between 1 and 10 days, either a single injection or a small number of injections, or a small number of days of once or twice a day oral therapy. And again, it needs to be a realistic dose that we can make, formulate, and deliver to patients. 40 mg per kg per day may sound like a lot, and I think that's very much the upper end. So DNDi is currently developing a drug, fexnidazole, which has a dose of 1800 milligrams per day. So in a malnourished patient, that can be, say, 40 to 50 kilograms. That can be getting on for 35 to 40 milligrams per kilogram per day. So, that's the upper end of what's possible, and it always brings challenges around formulation, synthesis, etc. But I think we would consider compounds with doses as high as that. Obviously, if that dose can come down to 10 mg per kg per day or less than that, it's going to give us a more chance of technical success in development. So that's the target candidate profile. To try and find compounds that would meet that, we've devised screen sequences. These are continuously updated, and different consortia working on projects have different takes on this, but this is a typical lead optimization screen sequence for us. As you can see, starting at the top and working down, we design and synthesize compounds, and then we'd put those into some in vitro assays, looking at Leishmania amastigotes in primary macrophages, and at the same time assessing cytotoxicity in those host macrophages, and perhaps also looking at a human cell line to give us an initial look at cytotoxicity against mammalian cells. If we find compounds with good activity, we'll then go on to look at initial in vitro ADME, so human liver microsomes, mouse and or hamster liver microsomes, because these are the species that we'll use for early in vivo efficacy studies, and also to have a look at solubility, lipophilicity, and permeability to help us understand whether we have compounds that could be administered orally, and if so, are we going to be able to formulate them? If that looks promising, we'll have a quick look early on at exposure following oral dosing, single dose oral treatment to a hamster or a mouse at 50 mg per kg, and to see whether we see exposure at an adequate level above, at least in the plasma, above the IC50 that we've measured in vitro. And if we do, then we'll take that compound forward for an in vivo study. At the same time, we'll look at the unbound fraction of the drug in plasma and also in the assay media to help us begin to understand total versus free drug, and to have that data in hand as we look at any emerging PK and PD relationships. We'd go on then to have a look at efficacy in a hamster model, or we could do this in a mouse model. And if at this stage very often compounds aren't working, we're going back through a cycle of design and synthesis to find better compounds here. If we find good compounds, then we'll go on and characterize them further, so looking at further in vitro ADME, further safety studies, as I mentioned in the target candidate profile, and also looking at our other animal models. So we'd go on to look at PK in a mouse, and I'm ready to run our second animal model. We'd also begin to look at activity against drug-sensitive and drug-resistant isolates and strains, so that we have a growing confidence about the in vitro activity of the compound against the different Leishmania parasites. For compounds that are really looking good, we'll go on and look at further cardiac ion channels, the profiling through enzyme and receptor panels, and looking at the drug-like properties of the molecule in terms of chemical stability, thermodynamic solubility, etc. If we can demonstrate good activity in one animal model, provided pharmacokinetics allow, we would expect to see good activity in a second model, and that just helps to build confidence that we have a good candidate, and also allows us to make an independent dose prediction to human based on a different species. If we've got down this far, we're probably very excited, and we're thinking about bulk synthesis, the early package for developing formulation, and getting ready to run an exploratory preclinical tox package, which for us would be 1 or ideally 2 weeks in rat, so that we can identify any major issues early before moving on into regulatory toxicology. So this for us would be selection of the compound, the optimized lead, before we move into the regulatory tox studies that were required as part of the preclinical candidate package. So that's a very fast journey through a complicated topic. That was my last slide. I'd be very happy to answer any questions that any of the audience might have.
Fantastic. Thank you very much indeed, Charlie. If people would like to pose any questions, please do. And we have one question, I don't know whether you can see it, Charlie, from Chris. How would you determine therapeutic index if efficacy was driven by Cmax rather than AUC? Oh, looks like I may have muted you, Charlie. Apologies. I don't think anybody will have heard that.
Okay, I'm back. Good question, Chris, thank you. For each new chemical class that we look at, identified from phenotypic assays, we don't know the mechanism of action and we don't know the PK/PD drive. So we have to be very pragmatic, and the initial PK characterization that we do is really to see, are we getting enough drug on board for it to make us worth doing an experiment to see if there's in vivo efficacy. If we get in vivo efficacy, then we'll do further studies to explore dose duration, dose level, frequency of dosing, to try and tease out what's the PD drive. Is it AUC? Is it Cmax? If it's AUC, it's probably simpler. We can compare AUCs from a tox study with an efficacy study. If we think it's Cmax, then I think you can start to have a similar discussion. You can try and work out whether any toxicity might be Cmax driven, whether it might be D driven, and start to have a think about what the therapeutic index would be. But I think it's a great question. We've presented the simple case, which is the simplest case, which is AUCs. But if it turned out that Cmax is the driver, you'd probably need to adjust those criteria accordingly.
That's great, thank you, Charlie. I'm going to suggest one more question. I've got one come in from Eamon. I would guess, Charlie, if other people do ping in some questions by text, you'd be happy to try and answer them briefly by email afterwards. Would that be okay?
I'll be happy to do my best. Yeah, I mean, I think you've got my email there, or you can share my email. I'll be happy to try and answer any other questions.
Fab, thank you. And so Eamon's question is, any general guide to the level of EC50 potency you would look for in initial phenotypic hits?
Yeah, another very good question. I think people are sometimes surprised at the apparently fairly weak level of potency that we're working on. But I think with our phenotypic assays, in the presence of protein, the free drug concentrations are probably lower than the apparent total concentration, and there's a number of those drugs that I showed you with really quite weak IC50s in vitro, so 10 to even 100 micromolar, so really weak, not nanomolar but micromolar, very weak. I think from our high throughput screening campaigns and our optimization campaigns, I can say it's quite challenging to find a compound with sub-micromolar IC50 against Leishmania. And if you can get down to 100 nanomolar or even less, you're doing really well. I think there's relatively few compounds that have a good selective mechanism of action that have that level of potency. So I think if you find a hit from screening that's in the 10 to 30 micromolar range, that's really not bad. If you can optimize that down to 1 to 5 micromolar, you've probably done quite well. And if you can get it down to less than 1 micromolar, then you're really competing very strongly with the best series that are available. Having said that, those compounds can still give really good in vivo efficacy at moderate doses in rodents and have reasonable human dose predictions.
Fantastic. Thank you very much, Charlie. So please keep the questions coming electronically if you'd like to. I'm going to just launch another poll that I talked about with Paul while I switch over presenters. So thank you very much indeed, Charlie, and we'll switch now to Paul Willis from MMV. And Paul and I discussed this question, which I think is often a hot topic in projects for infectious diseases. So we'll give you another minute or so. About half of you have voted. Okay, last 10 seconds to pick one or the other for the last few percent of you. Okay, I'm going to close the poll and share the result. So about 90% of you voted, and as you can see, it's a pretty split audience in terms of the opinions. Interesting. I will now hide that so that we can see Paul's slides.
Great. So can you see my slides and hear me?
Not yet, actually. I think I'm wondering if you've clicked the blue button. It should be towards the top left of your GoToWebinar panel. Show my screen. That's it. Perfect.
Okay. Did that work?
Yep. Lovely. Thank you.
Okay, great. And you can hear me fine, Mark?
Yes. Thanks, Paul.
Okay, great. So hello to everyone. Paul Willis from MMV. This is the catchy title I was given for today's talk. So I'm not going to talk too much about malaria, the disease. Really want to talk more about our target candidate profiles, drug discovery, and sort of bring out some learnings in terms of where projects have been successful, what's worked well, and some of the actual challenges. So in terms of our target candidate profiles, we actually start with the product in mind and what are the products that we want to launch to the patients. And really we're aiming for two different products. One you can see here on the right would be a chemoprotection product that would be used in areas where we're trying to eliminate malaria, where they would be treated with the drug to protect them from getting infection. So here this would be killing the malaria parasite either in the liver before it breaks through into the blood or the blood phase, and we call that the single exposure chemoprotection. Or on the left hand side, this is the treatment product we want to launch. So this would be a single exposure radical cure prophylaxis, or CERCAP as we call it. Current malaria treatments are given over 3 days, and we want to move to a single treatment. So that's the product in terms of treatment we're looking for. And then over the top of that, we then overlay, what are the target candidate profiles that go into those products. And these products will always be combination medicines to avoid the risk of resistance. So you can see here we number them, then we have 4 main target candidate profiles in this protection. In this treatment arm, we have a compound that needs to rapidly clear the parasite, this TCP-1, to give rapid release of the symptoms. And then a long-acting component, what we call TCP-2, that would provide protection for reinfection. then ultimately we want drugs. This 3A in the bottom left-hand corner, this is drugs that will eliminate the dormant form of vivax malaria that sits in the liver, phenomenally challenging for us. And then 3B would be compounds that would actually block transmission. And so treatment will be at least 2 different products, and they could share several attributes. So you might have one molecule that is both a fast killer of the parasite and then also blocks transmission. Within that, we can then sort of define the actual biological attributes that we have. Really, I just want to highlight 2 things on this slide. You can see at the bottom, for each of these target candidate profiles, we can show what the gold standard is. So for fast clearance of parasites, the artemisinin. So teams have a clear idea of what it is they're going to be. But I really just want to highlight as well just how challenging some of these target candidate profiles we're going for here. You can see here for the long duration compounds, then you can see where I've underlined this MPC, the minimum parasital concentration. So this is the minimum concentration to give the maximum rate of killing. We believe for efficacy you need to be above that concentration, and we want compounds that will stay above that for 8 days. So, you know, hugely challenging TCPs. And you can see similarly on the right-hand side for the chemoprotection product, you know, ideally we'd like once monthly treatments, but once weekly is the minimum. So, you know, really tough standards that we're setting here for new compounds. We recognise that those are phenomenally challenging, so those are our real aspirational goals. And then for each target candidate profile, we've set what's the minimum essential characteristics that would have to be met for us to consider a compound as a potential drug candidate, and then what our ideal is. And I've just picked out here for our fast killing, our TCP1 profile, and you can see here, just if you look at the dose, so our minimum acceptable would be up to 3 doses, a total of 1 gram. But what we'd like to move towards our ideal would be that oral one single dose of less than 100 mg. And I think this gives clear criteria that the projects know what they're moving towards for clinical candidates. And I think also what we're seeing is that, you know, quality of candidates has increased over time. So we're moving closer towards the ideal, and this can be a frustration to groups. So what might have looked a really high quality candidate 8 years ago can now look quite average today as we've built up a stronger portfolio of drugs. So I think that's a real positive about the area and having a strong portfolio that we're moving towards higher quality candidates. But obviously that sets the bar higher for projects in terms of designing new candidates. What really transformed malaria drug discovery was this move back to phenotypic screening, where we do high throughput screening looking for compounds that actually kill other malaria parasites within blood. It can be automated, and it's delivered a lot of leads. And you can see here, you know, we've now got 12 candidates delivered, 10 of which are still in either in clinical or preclinical studies. So this works really well. Most of the projects run as phenotypic design projects, but then by then generating parasites that are resistant to those drugs and sequencing them, you can identify the biological target. And what's interesting is that usually highlighted targets that hadn't previously been thought of as potential candidates. So this phenotypic screening has opened up new opportunities. So now we've actually deconvoluted the structures of many of those phenotypic hits, and I've just presented them here. You can see that we've got a load of targets that we know about now for that kill the malaria parasite. So when we're starting new projects, which are often phenotypic, it's important to know, are these working on an existing mechanism, or are they ones that we already know about? And you can see here, the third line down for the mechanism ATP4, we've now got 4 candidates. So probably we don't need at present more candidates against this particular mechanism. So understanding whether your phenotypic hit has a novel mode of action or or fit into one of these existing mechanisms is very important early on, as is that understanding of which target candidate profile your drug is going to fit into. And really to drive this, we now have the ability to screen against all stages of the malaria life cycle, as you can see on the slide here. At the top, where the malaria is infecting the patient by the mosquito bite, then going into the liver, moving through the blood, and then breaking out to the sexual stages, which are taken up in a new blood meal. So with new hits, we can screen against the whole life cycle, really understand what the biological properties of these hits are, and that really helps projects to know which target candidate profile that they're addressing very early on. And also within that then helps us to understand whether these might be new mechanisms or not. That the biological profile is associated with the mode of action. So you can see on this slide, we show in the 3 circles compounds that act on the blood at the bottom, those that act on the sexual stages on the top left, and then the liver on the right. And so you can see the intersection in the middle. We know about these 6 mechanisms which hit all stages of the malaria life cycle. So if that comes up as the biological profiling, then we've got that short list of potential targets that we can do further deconvolution to understand, do we think this is the biological mode of action? Yes. If it is another, say PI4K, is that something we want in our portfolio, or is it novel, in which case that would also be featured in. So this profiling really helps us understand where projects fit from a target candidate profile, and also where they fit into that broader mechanistic understanding. Then once projects embark on their optimization programs, we have the standard sort of progression through a drug discovery, from active to hit, through lead optimization to candidate. And at each stage, we've set quality standards. So you can see here, for example, early lead would be the entry point into lead optimization. So while we work as a virtual organization, all projects know and work to similar standards in terms of these are the criteria that need to be achieved in order to move a project into lead optimization. And these, the early stages of the hit to lead, these are decisions that are made by projects as we move through. For example, when we go to preclinical candidate, that would be made, a decision to approve there would be by our whole scientific advisory committee. And I just picked up 2 points here. You can see in this late lead criteria, in terms of, we're looking for really high potency against the parasite better than 10 nanomolar and good solubility. And I've just flagged those up as those are things I'll talk slightly more about as we move through the presentation. So as I say, most of our projects are phenotypic. And so we are then optimizing them based on a whole cell primary screen that in general is the red blood cell infected with the malaria parasite. And that can be a challenge. Some people question how easy it is to actually optimize on whole cell potency. Obviously, you're not measuring the true interaction of the compound with its molecular target there, and that cell potency can be influenced by permeability, protein binding, transporters, other effects. So perhaps when it started, there was a dogma, how easy would this be? But it actually works pretty successfully in malaria. I think one reason we're quite lucky is that the malaria parasite introduces new permeability pathways into the parasite, so you don't see those issues as much for us. So we can very successfully optimize on these phenotypic cell-based screens. I think something to be cautious of, and perhaps that we need to pay more attention to, is when we say looking at liver, blood and sexual stage screens, that all may be whole cell screens. They may run in different media with different protein content. So I think to be mindful when you're comparing across those values what the influence of that protein is something that's important, and perhaps we need to be paying more heed to in the future. I wanted to move on now, and then once you've started the project, talk about the sort of screening cascades that people might use. And on the right-hand side, I show what is probably a more classical screening cascade that might be used within pharma industry, where you may have a primary screen that looks at your potency on your known target, and then also runs a whole cell assay as well, and then would move into a whole battery of in vitro assays, and then the good compounds from that would move into PK, and you might do rat PK to understand the true PK. You might do mouse PK if your efficacy model was in mouse, and then move forward into your model. But the situation that we tend to address, that we run a much leaner screening cascade, and part of that is, we don't have the resource that big pharma have. We have to use a leaner cascade. So we're typically screening as a whole cell assay as our primary screen, and then we may move into a more limited evaluation of perhaps a solubility and perhaps rodent and human microsomes. And often from there, we will move for compounds that look as if they have good metabolic stability straight into an efficacy model, perhaps just a single dose where we'll measure both biological effect and then the concentration, and then really use the rat IV and oral on the side. And that's quite a different way of working. There's obviously a strength in the right-hand way where you have a real richness of data, but I think we've shown that this linear screening cascade can work very effectively, but you need to think very carefully about the decisions that you make, and because every screen costs money, then it's, what am I really going to learn from this screen? How am I going to use that? And I think this is something, I struggled to adjust to when I first came into MMV, being used to this very data-rich pharma model. But providing you think through your data and make careful decisions, this linear screening cascade can work very effectively, as demonstrated by the number of candidates that we do have in our portfolio now. So in terms of addressing these target candidate profiles, what are the features that it's useful to focus on? And potency is amazingly important. So projects that focus on potency tend to be successful. Simplistically, increasing potency can help to increase the dose, and low doses are beneficial when we're trying to treat small children who are very sick. And also, as we talked about before, for this long duration target candidate profile, the time above this minimum parasitic concentration is critical. So just this very simplistic graph, and this is a very simplistic analysis, I accept, but if you consider two compounds which have identical PK but have different MPCs, you can see the two lines, one for 5 nanomolar, one for 1 nanomolar. The lower potency compound maintains cover over that potency for a much longer period of time. So, we're not thinking about safety or other effects, but potency can really help you in terms of getting the dose down, and with other features extending the cover. So a focus on good potency is very important. But potency isn't everything, and some projects that struggle perhaps focus too much on just pushing potency down, and it's that classic medicinal chemistry issue of understanding the real strengths and weaknesses of your series, and making sure that you're focusing on all the key issues when you move forwards together. And questions like, if you're driving up potency, is the way that you're doing that actually having a detrimental effect on metabolic stability? So for us, the projects that have been most successful probably are these ones that really master this multi-parameter optimization. And from that, I just really wanted to stress the importance of log-D in driving a lot of these properties. And just to show that, here's some data on the slide here showing the influence of log-D on properties. Log-D is known to affect many properties: solubility, metabolic stability. So keeping the log-D in the right range can really help get your optimizing going. So here you can see the green box really shows, so on the y-axis, the actual values that you need to target, and then the green the sort of log-D range. So you can see by keeping the log-D below 3.5, you're getting improved herd potency, rat parasite stability, and human microcycle stability. So projects that do this sort of analysis, that really understand the correlations in their series, are often the ones that are most successful. And then this can be tremendously powerful. So here, for example, the project recognized that a log-D of less than 3 was giving them the best properties. So would then predict the lipophilicity of all new targets and use that to prioritize the ones that went through into synthesis. Again, synthesis resource can be more limited than perhaps big pharma, so really making the right decisions to focus on the key compounds to make. And here's just another example where really showing how analysis of your series and the SCR can really be enlightening in terms of information. So a plot of PIC50 against log-D and then just color-coded by substituent. So you can see in red, these are side chains that didn't contain a hydroxyl on a particular heterocycle, a reasonable correlation with log-D. And then in yellow, compounds that then that side chain has hydroxyl. And you can see for a given log-D, then these compounds do give a slightly bigger increase in potency, so suggesting that that substituent is beneficial. And then the team really used that to understand what was driving them forwards. And then interestingly, this set in green, where they either had an SO2 or a sulfonamide, there you have no correlation with log-D for that particular substituent. So that allowed them to move into a series where they could break this log-D correlation and move on. So really analyzing your series to understand the subtleties of what's really driving the properties are there, by breaking them down and looking at the effect of substituents, that can be really enlightening. So projects that are successful have tended to do that. Really, I just then wanted to end by showing an example where this has worked quite well, and this is a project, these are the early hit to lead phases from a project from the University of Dundee that was published very recently. And you can see on the left-hand side, so the initial hit compound, you can see in the top column, this is the potency, so a pretty good potency compound at 120 nanomolar, but a high log P at 4.3, giving what's not here shown, but it was poorly soluble, and you can see the next column down, the mass stability was poor. So the team straight away focused on reducing that log-D, so replacing the lipophilic bromine with the fluorine. You can see it has caused a slight loss in potency, but the log-D has decreased, sorry, we've now got a bit of solubility. And then using that now fluoroquinoline substituent to now look at changing the pyridine ring. Again, going to this prolidine ethyl side chain. You can see at this stage the potency is actually going in the wrong direction, so we're making things less potent. But the overall balance of properties is improving, so the log-D has stayed the same, but now with this basic group, solubility is improved and metabolic stability is now improved. So just to focus on potency might have missed this as an important breakthrough, but that understanding of the overall properties that counts, the team recognized that this actually was an advance, and then focused next on replacing the lipophilic phenyl ring. Again, with these side chains, again, you can see now then substituents of that position are maintaining or increasing the potency, and we've now got all green with a good log-D range, good solubility and metabolic stability. So this compound on the right-hand side then was the early lead declaration that the project moved forward. You can find more details in the paper, but that multi-parameter optimization was really what allowed that project to move forwards, really very rapidly. And I think there were only sort of 2, perhaps 3 chemists on the project, so a small focus team. So just to really summarize, I think by having a clear set of TCPs and progression criteria, that gives consistency and clarity across what is our portfolio of projects. we can successfully undertake phenotypic optimization of projects. And really, I think the key to success is this: maximizing the information that you take from a minimal screening cascade, really focusing on understanding what the data is telling you. So I'll finish there and be very happy to take any questions.
Fantastic. Thank you very much indeed, Paul. One question's coming from Tom von Gelden. Given the desire for very long half-life agents, do you consider biologics?
At the moment, we don't. Our specific focus is a small molecule focus. So it would probably be another organisation that would have to look at biologics. I think the work that has been done, it has seemed incredibly challenging, but perhaps there is a niche there for further investigation.
Okay, thank you. I just wanted to finish up in the last minute just to show people that you can access information about this series of presentations from MMV's webpage. If you go to the computational chemistry page, which MMV have been very kind in hosting for us, if you click on this link here, the see information about upcoming meetings and recordings of previous meetings, you do have to click through a couple of times, but what you get to is basically this Word document which we maintain, which has a link to the recordings of previous presentations. That is when I remember to hit record, which I did partway through Charlie's presentation, and also agendas for upcoming meetings. So you can see a brief advert for the one on mid-March. Isabelle Giraud, some of you may know, she did the first one and didn't. There's some more interesting stuff that she wants to talk about in terms of Data Warrior, and Andy Davis talking about his RSC medicinal chemistry toolkit. And then the one in April will show you some of the amazing things that the free tool NIME can do for you. So we just check. Excellent. I'd like to just conclude by thanking everybody for attending today, and thanking in particular Charlie and Paul. And you will get links to the recordings and some of the other answers that Charlie's given to a couple of the questions that came in during the session. So thanks once again, everybody, and hope to see you on the 17th of March. Thanks a lot.